Hello, this is Parashar Vikas, Block 8, and here I am presenting urea cycle. So, what is urea cycle? The conversion of ammonia into urea through a series of biochemical reaction is known as the urea cycle or ornithine cycle. It takes place in the liver with the help of mitochondria and cytosolic enzymes. It is an import pathway in amphibians and mammalians as they help in disposing highly toxic ammonia by converting it into urea. We cannot excrete ammonia directly, so they convert it into simpler form of urea. The urea converted in the liver is transported to the kidney by the bloodstream and then finally excreted in the form of urea. This process is important because if the nitrogenous waste is not excreted, it start building up the body and can be determinantal. Urea is insert in nature, soluble in water and can be easily excreted in urine, whereas ammonia is highly toxic. So this is urea cycle. Next I will explain the steps of urea cycle. So these are the steps. So the urea cycle start in the mitochondria of the hepatocystic and the final step take place in the cytoplasm. The final product form is then transported to the kidney, whereas it is excreted out of the body. Entry into the urea cycle. Ammonia and carbon monoxide are converted into carbamoxyl phosphate. In this rate limited step by the enzyme carbonyl phosphatase synthesis. 1. 2 ATP molecule are utilized in this step. Ammonia becomes the source of the first amine group in urea. The CPS1 require an obligate activator, namely N-acetyl glutamate. NAG is formed by a reaction between glutamate and acetyl coenzyme in the presence of NAG synthesis. First step. In the first step, carbamoyl phosphate enter the urea cycle and combine with ornithine to form citrulline in the presence of enzyme ornithine transcarbamylase. The citrulline form is then transported out of the mitochondria into the cytoplasm by ornithine transloop. Second step. Citrulline and aspartate undergo a condensation reaction to form arginosuccinate in the presence of enzyme arginosuccinate synthesis. Here, aspartate becomes the source of the second amine group on urea. This reaction utilizes one ATP molecule. In the third step, arginosuccinate is cleaved off to make arginine and fumarate in the presence of enzyme arginosuccinated liase. The fumarate is used in the production of NADH in the TCA cycle, and the arginine move forward for the next step of the urea cycle. The fourth stage, arginine undergo hydrolysis to yield urea and ornithine in the presence of arginase. Ornithine is transported back to the mitochondria, which is used up in the second step of the cycle to form citrulline by combining with carbonyl phosphate. The overall reaction equation of the urea cycle is NH3 plus CO2 plus aspartate plus 3 ATP plus 3 H2O gives urea plus fumarate and 2 ATP plus 2P plus 1 AMP plus PP plus H2O. Regulation of the urea cycle. Uh, N-acetyl glutamate in the urea cycle. The enzyme CPS1 is allosterically activated by the NAG. It is an obligate activator of the carbonyl phosphate synthesis. NAG is produced by the reaction of acetyl coenzyme and glutamate in the presence of NAG synthase. And the reaction is stimulated by both arginine and glutamate acid. Therefore, glue is both a substrate and activator for the urea cycle. Substrate concentrations. All the enzymes in the urea cycle work based on the concentrations of their substrate except arginine. So these are the disorder of urea cycle. Ornithine transcarbamylase deficiency. It is the only X-linked recessive enzyme deficiency disorder in the urea cycle. The deficiency of the enzyme leads to an increased concentration of the carbamyl phosphate in the mitochondria. The carbamyl phosphate is channeled to the pyramid synthesis pathway in the cytoplasm. It is then converted into orotic acid. Orotic acid is built up in the blood and urine and appear as orange crystals in the diaper of an infant. Diaper of infant. Arginosuccinate synthesis deficiency. Arginosuccinate synthesis deficiency, also known as the citrullinemia type 1, is an autosomal recessive disorder of the urea cycle. This disorder is associated with buildup of citrulline and ultimately ammonia in the infants. The disease is represented by seizure, lethargy, anorexia, and respiratory distress. In the adult, it appear in the milder form of symptoms include vomiting, ataxia, and lethargic. Carbamoyl phosphate synthesis 1 deficiency. It is an autosomal recessive metabolic disorder that is commonly seen in infants. It makes the child lethargic, unwilling to eat, altered body temperature and respiratory rate. In the other reflected individual, this is shown symptoms only later in life. This was all from my side. Thank you.